afternoon. My name is Joe. I'm a CEO and founder of a company called mbiomics. And we do microbiome-based therapeutics. So that means we take live bacteria, put them into a pill, put the pill into a patient, and these bacteria engraft in the microbiome of the patient and thereby have a therapeutic effect. And there's a couple of things I want to give you in the next couple of minutes. The first is I want to convince you that the microbiome is going to change completely how we do therapeutics in the next couple of years. And the second thing is I want to convince you that this is only a technical challenge. It's an engineering challenge to get the microbiome right. There's no more magic required. So let's get started with some microbiome basics. The microbiome is a really exciting organ in our gut. We have each and every one of us has a couple of hundred bacteria living there, a couple of hundred different species, 30 trillion cells in total. That's more cells than we have in the human body. And it's a very complex ecosystem. And this ecosystem is very different in between all of us. So understanding the microbiome in the first place, understanding how a functioning microbiome works and how that correlates with disease is essentially a computational problem in having a very large data set that you need to understand here. Yeah? So we talk about sequencing a microbiome, getting about 4 million genes out of that sequencing run. And these are genes you've never seen before. You don't have a database to look up what they're doing. So really in understanding a microbiome, you need to make prognosis on new genes, what kind of enzymatic capability they might have. Now, if you're able to tackle the microbiome, there's a whole suite of indications you can attack with that. The microbiome is closely correlated with the immune system, but 70% of your immune cells are located around your gut and monitor what's happening there. You have a very close link with the gut-brain axis to hormone and neurotransmitter release, and obviously there's all the metabolic effect you're having. So if you can tackle the microbiome, you can tackle all these diseases. This is no magic required anymore. This is this is very hard science we have here. The science we get are from hundreds and thousands of studies with fecal transplants. So a fecal transplant is a healthy person donating a stool sample, and that stool sample is transplanted into a patient. And this obviously is a very powerful academic tool and something that you can also get into the market if you're really pushing for it. But it's very tough to commercialize because it's not scalable. Every donation is going to look different from donation to nation, from donor to donor. It's very expensive to do. So what you really want to do is not manual lab work for every donation, but you want to have a scalable process in a fermenter where you grow these bacteria, right? Because growing bacteria is an incredibly scalable process, very efficient, very low cost of goods. So the engineering challenge really comes down to being able to take a consortium of bacteria, make that as efficient and as effective as a fecal transplant, so very complex, but be able to reproduce that in a bioreactor. And that's something we've been doing the last couple of years, building a technology stack of doing something like that. And we've been looking through the indication space on what kind of disease could you tackle with, as a demonstrator first, and we decided we're going into oncology. We're building an immune oncology drug that helps immune checkpoint inhibitors to kind of double the response rate they're getting in patients. So instead of only a third of the patients getting this revolutionary immuno-oncology treatment to be efficacious, you suddenly get 2/3 of the patients. So how does this work on a product side? We're doing 150 selected bacterial strains. We designed them in silico to put them into a pill. And there's kind of 3 different modules we've been designing in here. The first, the large orange bubble, is we're designing a fully functioning microbiome, a synthetic microbiome here. These are about 100 strains that are selected to have a very high taxonomic breadth. So we're really making sure you're capturing all the different variances in taxonomy, but they're also tailored to each other to have the full enzymatic cascades, the metabolic cascades, to really get the effect of a full microbiome. And then there are 2 more modules here. One is the mode of action specific, and the other one is indication specific elements. So we're making sure we're looking at the bacteria we really need to make an effect in a certain disease and get the right metabolites we need for that disease. So especially in the immuno-oncology, it's very clear what you need to trigger in the immune system for an immune checkpoint then to respond. That brings us to, as I said, 150 strains, and this is obviously a huge challenge in designing what strains to pick, to manufacture them, to analyze them. And I've been just giving you a bit of a high-level rundown on the platform we've been building. This is a AI-first problem, right? You need to understand how the microbiome works. I talked about that very briefly. But then you also need to design the product and understanding which 150 strains should you actually select to get the functional profile you want. It's also a biological problem then. You need to have these bacteria in your freezers. So we've been isolating bacteria from humans, from healthy donors. We have something like 14,000 characterized strains in our freezers to select from. And then there's the next challenge. You have the stuff in your freezers and you have the design you would like to be built, and you need to map those two and make sure you're picking the right things. From the strain library, we're going into the co-culturing here. And co-culturing is a very important thing because you can't just manufacture 150 different strains and then pool them together afterwards. This is prohibitively expensive, also logistically really complicated to have 150 GMP runs. So what you need to be doing is you need to grow these bacteria together. And everyone who's been doing some microbiology here knows it's quite complicated to grow strains in the first place. 15 years ago, a guy next to me in a bench made a PhD thesis on growing 2 bacteria together at the same time. We say we do 150 strains. That is powered by analytical technologies. You need to understand how these bacteria grow together at all different time points. You need to understand their relative abundance. You need to know whether they're alive. You need to understand how they behave in an animal model, how they behave in a patient. And that obviously also gets you to screening. You need to understand the stuff you've been designing in silico. Is this really happening in a biological system? Are the cells responding the way you want? You need to have mouse models that mimic a human, so you can really make a prognosis on that. And for the last 2.5 years, we've been building this platform. We've been really starting with a blank piece of paper saying this is going to be a probably 10-year journey we are facing in getting a microbiome company on the market and staffed with technologies. We've been looking at what kind of technologies we need to develop and get into the company, and we are something like a fourth on the way. As I said, we started with immuno-oncology as the first demonstrator, and putting this on a timeline here, we've been building platform, getting our MBX 116 product into the preclinic now, getting ready for manufacturing, and then early 2027, we're going to get this into the first humans in our first clinical trial. We've been designing this, I think, quite aggressively. We are manufacturing now while we're doing the mouse models to really pick up speed on the development here. And we plan to have here mid-2028 already efficacy data on that product to really make sure this actually works. And we're now designing the next pipeline products here where we'd love to go into autoimmune diseases, so we're looking for partners in that. So what we've been doing the last couple of years is a 20 million round to fund this really from scratch to get going. We're currently getting some more money into the company to be a bit faster, do a couple of more things in parallel, and to run a clinical trial. We're looking for a 50 million round, and I think that gets us to why I'm here. At mbiomics, we're very curious about awesome people. So if you want to talk about stool samples, maybe more the microbiome, if you're a scientist, please come to me, talk to me. I'm going to be spending some time behind the stage here. If you're an investor, we need really excitable people to follow through with this the next couple of years. And so please get in touch and say hi. Thanks a lot.